What if you could solve a problem like allergies, not with a pill or a shot, but by editing the very code of life itself? Well, that's not science fiction anymore. It's the real story we're diving into today. A story about science, some very special cats, and a truly revolutionary solution to a problem that affects millions.
I mean, it's a super frustrating reality for so many people, right? You absolutely adore cats, you want that furry companion in your life, but your own body just rebels against the idea. It's a classic dilemma, and it's one that scientists have decided to tackle head-on.
And we're not talking about a small number of people here. In some places, up to 30% of the population is allergic to cats. That is a huge number of people missing out on having a cat, which makes the hunt for a real, permanent solution just so important.
Okay, so first things first. To solve the problem, scientists had to find the exact culprit. What is it really about cats that makes us sneeze and itch? Well, it turns out the answer is microscopic, and it's incredibly sneaky. So, meet our microscopic villain, FeLD1. This tiny little protein is the main troublemaker. In fact, it's responsible for up to 90% of allergic reactions in people who are sensitive to cats. So get this. It's not really the fur you're allergic to, it's this little protein that hitches a ride on the fur.
And this protein is a real nightmare for allergy sufferers. Why? Because it just gets everywhere. It floats in the air, it's super sticky, and it can hang around in your carpets and on your clothes for months. You basically can't just clean it away. That means the only real way to fix this is to go straight to the source, the cat's own DNA. And this is where things get really, really cool. To fight FeLD1 at its source, scientists needed some seriously powerful tools from their genetic toolkit. Let's take a look at the two key pieces of tech that made this whole thing possible.
First up, we have cloning. And you know, this isn't just something out of a sci-fi movie. It actually happens in nature. That's what identical twins are. In the lab, it's a technique that lets scientists make a perfect genetic duplicate, and you'll see why this is so important in just a bit. The second tool is a real game changer. It's called CRISPR-Cas9. The easiest way to think about it is like a find and replace function, but for living DNA. It works like a pair of tiny, programmable scissors that can go into the DNA, find a very specific gene, like the one that makes FeLD1, and snip it right out, or change it. It is the ultimate gene editing tool. So, how does that lab-based cloning actually work? Well, the process is called somatic cell nuclear transfer, or SCNT. It sounds complicated, but here's the gist. Scientists take a regular body cell from the animal they want to copy. They pop out its nucleus, that's where all the DNA is. Then, they take an egg cell and remove its nucleus. Finally, they put the first nucleus into the empty egg. That new egg can now grow into an embryo that's a perfect genetic copy of the original donor. Okay, so we've got our problem, this pesky FeLD1 protein, and we've got our high-tech toolkit, CRISPR and cloning. Now let's see what happened when scientists put them all together to create the world's very first gene-edited hypoallergenic cats. This is where it all comes together. All right, let's meet the pioneers of this story. On the left, we have Heavy, and on the right, Hamey. Scientists used that CRISPR tool to go in and edit the gene responsible for FeLD1 in the cells that would eventually become these two cats. This made them the founding parents of a whole new hypoallergenic family line. And this is their amazing offspring, Alsik. Now, when Heavy and Hemi were bred, Alsik inherited that edited low-allergen gene from both of his parents. And that's the key. It means this trait is totally locked into his DNA. But of course, the big question was still hanging in the air. Did it actually work? And the results? Well, the data speaks for itself. Just look at this chart. A regular cat's saliva had almost 11 micrograms per milliliter of the FeLD1 protein. Alsik's? A minuscule 0.15. That is a staggering reduction of over 98%. The gene editing didn't just work, it was a home run, an absolute success. And hey, don't just take my word for it. The scientists themselves said it all in their research paper. They wrote that Alsik's allergen level was extremely low. That wasn't just some small adjustment. This was a fundamental change they had created. But you know, in science, doing something once is amazing, but it's not enough. You have to prove you can do it again. It has to be repeatable. So the team needed to deliver an encore performance to really prove their point. So you have to ask, right? Was Alsik just a fluke? A 1 in a million lucky break? Or had the scientists truly figured out a reliable way to make hypoallergenic cats on demand?
They needed to prove it, beyond a shadow of a doubt.
So, they cloned him. Meet Alsik C. Using that same SCNT cloning method we talked about, scientists made a perfect genetic copy of Alsik. And the result? Alsik C also had those incredibly low levels of the FeLD1 protein. This was it. This was the final proof they needed. The hypoallergenic trait was real, and it was replicable.
And to really appreciate what a big deal this is, let's just put it in context. The journey started back in 1996 with Dolly the sheep, the first mammal ever cloned. Then in 2001, we got CC, the first cloned pet cat. And now, Alsik C represents the next massive leap forward. Not just cloning an animal, but cloning an animal with a specific, beneficial, engineered trait.
Okay, so a cat you're not allergic to, that's amazing, but the story, it gets so much bigger than that. The technologies we're talking about here have implications for the future of medicine that are just profound.
Now, this is a really important point to get. The process used to make Alsik C is called reproductive cloning. The goal is pretty simple: create a whole new organism. But there's another kind called therapeutic cloning. It starts the same way, but instead of making a whole being, the goal is to create specialized stem cells that can be used to treat diseases.
And the potential here, it is just mind-blowing. We're talking about maybe one day generating brand new neurons to treat Parkinson's disease, or creating insulin-producing cells for people with diabetes, maybe even repairing spinal cord injuries. It could lead to cures for genetic disorders, and down the line, maybe even growing replacement organs on demand perfectly matched to a patient.
So yeah, it all started with a sneeze. And it's led us right to the edge of a brand new medical frontier. The quest for a hypoallergenic cat has unlocked tools that could literally reshape human health as we know it. So the only real question left is, what do we build next?